Hey there, everybody. Welcome to Microbiology. This is Chapter 1. We use the Microbiology, A Human Perspective by Nestor Anderson and Roberts. It's the 7th edition. If you have an earlier edition, it's not the end of the world. Editions change minimally typically, okay? But with this textbook, we have a student website that you can go to for extra help. And so I'm going to show you this website really quick as soon as it loads up. Here it is. In here you have pre and post tests. There's image PowerPoints. There's animations. You can go to Chapter 1, and in Chapter 1 you can take video or animations. Here's a 3-domain animation. Here's a life cycle of the T2 phage, which is a virus. Sometimes I'm going to use these tests here to pull some questions for the exam. So you definitely want to go into this companion website because it's really beneficial. There's all the different chapters. So whatever chapter we're in, you can go to, okay? I will be posting all of the notes and your PowerPoints on Moodle, so you'll be able to find them. Any homework assignments, disease assignments will be posted on Moodle as well. I will also be uploading your grades to the Moodle gradebook, so you should always be able to tell what your grade is in the class. Just for your information, if you forget who I am, I'm Krista Winters. My email address is kwinters@sandberg.edu. You can click on this and it'll bring you right to the email, okay? If you have that email, I should say. My office hours are by appointment only for, well, 2 reasons. It's summertime, and so in summertime we don't have scheduled office hours, but also because I'm part-time and I'm adjunct here, so I don't actually have an office. Typically, my office hours for you will be right before class or right after class. If you need me, come in. I will be on campus. I teach a general biology class from 2 to 4:30. And then from 4:30 to 5:30, I will be around possibly prepping for your class. But if you let me know you need to talk to me, I will make sure I'm there for you. So let's get into Chapter 1. Chapter 1 is all about what microorganisms are, the importance of microorganisms, and why we study microorganisms and how we study microorganisms. So it's a lot about history of microorganisms. Microorganisms are the first life forms on Earth. They have been dated back about 3.5 billion years. So the Earth is about 4.6 billion years old, and microbes have been on the Earth for a good portion of that time. Whereas animals and plants have not been around until, so if you're looking at plants and animals, we're looking way up here about 500,000 years ago, okay? Humans, you know, just the last few drips on the time scale. So how did microbiology get its origins? I like that cartoon. It kind of reminds me of the importance of microscopes and microscopy. The study of microbiology, microbiology is the study of microorganisms, and we have to have certain tools to study these microorganisms. So but prior to the production of these tools, we couldn't study the microorganisms, and we probably didn't know, we didn't know that, or what microorganisms were. And I just thought this was cute. I don't know what this is, but you should see how fast it's growing. Rawr, he's gonna eat him. See, kind of funny. So we had to make microscopes so that we could actually see microorganisms. we had to come up with techniques so that we could grow the microorganisms effectively. And then we came up with just recently, as recent as we started figuring this out in around the 80s, the molecular genetics of the microorganisms. So microbiology was born in the 1600s. Robert Hooke was one of the first individuals to produce a microscope. And he made a microscope because somebody, I don't remember who, but some wealthy individual wanted someone to look at the microscopic world. And so Robert Hooke said, I'll do this. And so he came up with a way to observe the microscopic world using a microscope. So this is his microscope. It's not a very strong microscope. It can magnify, tens of times compared to what we magnify at hundreds of times. But he was able to see, when he looked at cork, these tiny little square structures that had like space in the middle, and he called them cells. And the name cell came from the rooms that he was staying in the monastery that he lived in. They were very tiny rooms, and they were close together, just like the cells in the cork that he saw. And so he coined the term cell, and he actually is one of the individuals that is given props for coming up with a portion of the cell theory. And the cell theory, all living things are composed of cells. Cells produce new cells, and we know this. He also drew some of the first cells, or some of the first microscopic organisms. So what he drew was what he called a microscopic plant, and we now know that that microscopic plant that he grew was actually Rhizopus stolonifer, which is a fungus. And so he was drawing this, and so he's one of the first individuals to see microscopic organisms. But he's not the first to see like protozoans or bacteria. So we move on to Antony van Leeuwenhoek. Leeuwenhoek was a Dutch drapist, and he wanted to make, or he didn't want to, he was making very fine drapes for some of the richer folks. And to make them very good, he used a lot of magnifying glasses to make sure that all of his stitches were perfect. Well, while he was doing that, he started thinking about what else he could see with the microscope, or with his magnifying glass. Because he wanted to, or because he was interested in looking at other things, he decided to take some pond water and look in it, and what he saw in the pond water were these tiny little microscopic animals. And so he called them animalcules. And he actually drew pictures of them, showed how the animalcules were moving. He then looked at teeth scrapings, and he saw other microscopic organisms. And some people actually think of Antony van Leeuwenhoek as the father of microbiology, because he was one of the first individuals to see living microbes. So now we know that there are microbes. So the question comes, or that arises from that, is where did these microbes come from? Most people thought that microbes and that all living things spontaneously appeared. They just showed up magically. kind of like the color just appeared magically. Um, we know today that life comes from life. There's a theory called the cell theory that states cells are the basic unit of life, and only cells can produce new cells. So life comes from life. But back then, the theory that most individuals believed was called the theory of spontaneous generation. What are you doing, Dudley? My dog is digging on our bed. That's weird. Anyways, so most people thought spontaneous generation was how living things came about. So microbes that were growing, like fungus that was growing on your food that had been set out or that had been set in a jar for multiple days, just spontaneously started growing there. There were no spores or anything in the food that germinated. It was just spontaneous. And so when I say that most people believe this, this was a theory, the theory of spontaneous generation. And so one example of that theory is if you have dirty clothes and you put those dirty clothing, so like cloths and stuff, in a dark, cool area where you keep maybe wheat or other grain, in a couple of weeks, you're going to have mice. Or, if you leave meat sitting out, eventually worms will start growing in the meat. They'll just spontaneously appear. So this is a theory, and a theory is in biology is really as concrete as a law in most other areas. When we have a theory, that means that we have tested and tested and tested the whatever the hypothesis was, and we have supported it multiple times. So the theory of spontaneous generation was something that scientists, almost all scientists, the church, and because most of the individuals that were in charge believed, most laymen also believed. And so I have a little video, spontaneous generation. All right. So let's look at the individuals that supported spontaneous generation. The scientists, the church, so most individuals. One individual specifically that I'll talk about is John Needham, who was with the church. And then we have a lot of detractors as well. And so these are the individuals we'll talk about: Francisco Redi, Lazzaro Spallanzani, Louis Pasteur, and then Tyndall and Cone. They weren't really supporters or detractors, I won't say that, but they helped with Louis Pasteur's, okay? So in 1668, Francisco Redi, he showed that if you took meat and you put it into two open jars and you left the meat sit, but in one of the open jars you covered it with a piece of cheesecloth, the only one that would have maggots were the ones that the flies could touch. And so what he said was the flies are laying eggs and the maggots are coming from the flies. So life is coming from life. And this really caused a lot of problems because the people that believed so heavily in spontaneous generation couldn't figure out why no maggots were forming here.
Mice emerge.
Another evidence offered for spontaneous generation.
So then Needham decided to run his own test. What he did was he said, okay, here, I'm going to make a broth with chicken infusion. So he took chicken, basically, he took chicken and put it into some water. He boiled it. He allowed it to sit. There was microbes growing in it. He boiled it to kill all the microbes, so there was nothing there. And then he let it sit, and of course, microbes were there after days after the broth was boiled. And so people thought, okay, we're not sure why this didn't work here, but we do notice that, hey, there's something going on here. And so they thought maybe the only type of organisms that could grow spontaneously, spontaneous generation, you had to be very simple, and maybe flies weren't simple enough, but microscopic organisms were. So they're like, it's still true. Spontaneous generation is still a theory that is true. So then Lazzaro Spallanzani said, okay, let me do the same example. He did it, but he used two experimental, he did two experiments at the same time. The same type of broth he put into two different beakers. He stoppered one of the flasks and the other one he didn't. Boiled them. There was no microbes in them. And then days later, there were microbes in the open flask. But in the unopened flask, there was nothing. And so he said the bacteria, or he said the microscopic organisms, which they called germs, were in the air and got into the infusion. And here they couldn't get into the infusion because it was stoppered. And people thought, no, 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 because you have this stoppered, there's a vital force in the air that has to be able to get in. So microbes might not be able to grow because there was no vital force here. So then Louis Pasteur came along and he was like, okay, let me figure this out. So the first thing Pasteur did was he showed that there were microscopic organisms in the air. And he did this by taking a flask and putting a cotton ball in there, and then he filtered air through the cotton ball. He is considered the father of modern microbiology. And when he filtered the air through the cotton, he showed that the microbes that were on the cotton ball were the same microbes that were found in infusions. And then he ran an experiment. He made a swan neck flask. So here we have a flask, and he took the neck of the flask and he bent it. So the bending keeps microbes from being able to move all the way through the flask. Microbes can't move like we do, so they can't get through those turns. But air could still flow in. And because of that, the broth, when it got boiled, the microbes that were there were destroyed and they never grew again. And so he could set this flask for weeks, months, even years, and nothing would grow. But as soon as he either broke the neck of the flask or he turned the flask so that the neck, so that the broth went into the neck here, the microbes would start growing again. And so air was able to get in, and so that means that the vital force wasn't there and nothing grew, which means that the theory of spontaneous generation had been debunked. Now, this is in the 1800s. So scientists are always trying to reproduce results. When you go to college and you're working on your undergrad research or you're doing grad research, you often start by doing experiments that other scientists have done in the past to, learn how experimental design and everything work. And so scientists would take Pasteur's work and then they would run the same experiment, but they didn't get the same results. And so if the experimental design is perfect, the results should be the same every single time. And so they weren't sure why they weren't getting the same results, and so they didn't agree or they were skeptical of his work. Well, then came along Tyndall and Cohn, who explained the discrepancies seen in the results that Pasteur had and the results that other scientists had. And it showed how important experimental design is. So John Tyndall came along and he started looking at Pasteur's research and then looked at other people's experiments to see how they differed. And what he noticed that was that you could take infusions, so Pasteur's infusions, and boil them, and after 5 minutes, life was gone, and no life would ever come unless you opened it to the air. But if another scientist used their infusions, that didn't happen. But he looked at the different infusions and he said, okay, you're using a different type of infusion. Your lab is set up in your barn, so not everybody had beautiful, perfect microscopy labs and stuff. So a lot of these individuals would just set up their lab in their barn. And if they had their lab set up in their barn, then a lot of times the microbes that were there in the barn in the hay were, excuse me, my dog's going crazy, were actually heat resistant. So they produce these little structures called endospores. And if they were heat resistant, if they produced endospores, then even if you boiled for hours, the endospores would persist. And as soon as everything cooled down and the environment became beneficial for life, the endospores would germinate and you'd have those bacteria again. And so that was why they were getting different results. And so he found that basically there were two different types of organisms, heat heat-resistant organisms, which are the endospores, and non-heat resistant organisms, which are the vegetative cells. And the non-heat resistant organisms die when you boil the infusions. The endospores survive. And it was Cohn who actually identified the heat-resistant microbes as endospores. And endospores are very commonly seen in barns because Clostridium often are found within cows or in cows and around cows in the dirt, in the fecal matter and everything. And Clostridium species are endospore formers. So this explained why there were differences from one experiment to another. If you don't use the exact same experimental design, the results will always vary.